Welcome and thank you to those of us joining to listen to our session on discussing issues around the intervals between the first and second doses of the COVID-19 vaccines. I am sure many of you are still waiting for your first dose, and we hope that that will be here soon for all of us. And I know many of you have also received your first dose and are now wondering about when you're going to get your second dose, and perhaps have some concerns around delays between what was initially intended and when you're going to actually receive that second dose. I'm Brad Waters. I'm the Executive Vice President of Science and Research at University Health Network, UHN in Toronto. And I'm pleased today to be moderating today's session. We know many of you have these questions, and this is really an opportunity to hear a little bit about the science and the thoughts that go into the decision-making between the decisions that had been made most recently around the extension and delays between the first and the second doses. Given how quickly the research is evolving and the really avalanche of information out there, we're really grateful to have two experts here today to explain the science behind the decision and to answer some of the common asked questions. Certainly, I've heard a lot of those at my own institution. And I want to just start by making a few comments around how I think it's important to frame the conversation really around two issues. The first is around the vaccinology itself and what the science tells us, why we give two different doses, what happens, how's the immune response involved in doing that, and how we might use the understanding of that science to help think about decisions or consequences of changing dosing patterns. And the second is really around more the principles of practicing evidence-based medicine. And of course, when in medicine today, we like to give treatments and to deliver therapies in ways that is based on good clinical evidence that is created through the practice of clinical trials. And that helps guide the way we deliver therapy. So I want to start off first by turning over to Dr. Lin to give us a bit of an introduction into the science. Dr. Lin.
Thanks very much, Brad. Thank you for those opening remarks. I think they're very useful for us. I'm just going to start off here. So let me just take you back a little bit. So how does these vaccines work and so on and so forth? I think many of us have heard about the mRNA. So that is a copy of the DNA, and that's the set of blueprints that goes to your factory to make any protein in your body. In this case, what they've done is they've taken the piece of the mRNA that will code for that spike protein, and they package it into this little lipid ball, and therefore you can actually take it into your body. And so that little piece will go to your factory, and then you will make the protein. So therefore, your body will make the spike protein. And once the spike protein is shown, then your body will make antibodies against that. So it's a very nice way of doing things. And the messenger RNA breaks down very quickly. So therefore, it doesn't hang around for very long. And once you've made the protein, you show your body. It's like a wanted poster. So therefore, your army and your immune system knows what the foreigner looks like, and therefore will make the antibodies appropriately. Now, I had a nurse friend that called me up, and this was during the time when all of a sudden there was an announcement that there was no more vaccine coming, and she was in a panic, and she's an experienced nurse, and she was in a panic because she said, I only got one shot, so that means I'm not going to get the boost that I need. And so that's what we traditionally think of. And we've been hearing numbers like the first shot gives you 50% protection, and then the second shot is needed to boost you to 95. And so therefore the sense was that she was being, disadvantaged, and at one point she was saying, I wish I hadn't even taken the first shot. And so instead of just, saying, okay, it's okay, don't worry, and in other words, just empty words, I decided I better go look up the study itself and see, can we learn anything at all from the graphs and the data that we already have? Because I know the numbers have been floating around, but what do the graphs look like? So for this particular one, this is the Moderna one, and you'll recognize it because it'll have this as the vaccine name. And so what you see here is the following. So this is the placebo group in terms of people getting sick with the virus, and this is the group that was vaccinated with the true vaccine. And what you could see here is that it's relatively flat. So we do have these wonderful numbers here, like 94% and so on and so forth. But what impressed me was this very flat line that was going out in this direction. So then I thought, okay, what about the Pfizer one? The Pfizer one looks very similar. Okay, so if you look at the Pfizer one, which is what many of us have gotten, what you could see here is that again, there's this sort of flat line that goes out, almost hinting as if people were not getting infected after a certain time point. So that gave me some interest in this, is that what happens during this time? So out to about 90 to 100 days, that line seems to be very flat and it doesn't seem to change much. So that was very encouraging that maybe these vaccines, number one, are working very well to get you with those antibodies to protect you. But then I was interested in what happens in the first 21 days because my friend had just received her shot and she was not going to get the second shot. So, what happened in that very first 21 days after the very first shot? And so they actually have a beautiful graph of the first 21 days. And what you could see here is that in the beginning, both groups, whether you got the real vaccine in red or whether you got the placebo shot, you could see that they get infected at the same rate. And presumably you haven't made the antibodies yet, so you don't have any defenses. But you'll notice somewhere around day 11, day 12, it becomes like a hockey stick, an upside down hockey stick, I call it. And so all of a sudden, it looks like the cases don't seem to be happening. So it looks like you've built up enough antibodies to stop the virus from giving you sickness. And so if you look at that, right here, it starts to go flat and then it continues to stay flat out to this area. Now, remember, they did get a second shot and we often give a second shot. And is the second shot needed to give you the benefit or is it to give you more money in the bank, so to speak? In other words, give you more antibodies, so therefore it'll last longer. And so if we look at the time frame of when they would have gotten it, it would have been around here after the 21st day out to, let's say, about 42 days or so. And you can see here that the line is flat. And the reason why I point that out is that if we really needed the second shot to give us the protection, the first shot would have reduced the number of infections, and then the second shot would have reduced it more. In other words, we would have seen some flattening of the curve and then further flattening of the curve after that second shot was given. But as you can see here, it actually stays fairly flat throughout. So what I'm interpreting for my friend is that maybe that first shot is giving you enough antibodies to protect you against invasive disease. In other words, the virus coming into your body and making you sick. And that perhaps that second booster is just giving you more antibodies. So therefore, this will last longer as well. And so that's why when we start talking about durations of when we should get the second vaccine, we know that 42 days, which is what the NACI recommendation has been, you could see that you're fairly well protected. And so therefore, that's quite safe. And you could see why some people might interpret this and say, look, the line is pretty flat out to about 90 days even. And so therefore, that might give some rationale as to why the UK and Quebec said maybe even out to 90 days is okay. Of course, if we can get the vaccine earlier, then we give everybody based on the traditional way of the proper duration. And we've also talked to some, immunologists, and they said, we always want to make sure that we don't give the vaccines too close, because if you give the booster too close, then you don't get the full effect. But if you actually stretch it out a little bit, you actually get a better boosting effect. So therefore, if there is a small delay, there shouldn't be any harm coming to you because you're already protected from the first one. And number two is that even if it's a little bit delayed, it might actually give you a better boost. Now, we do have to remember that in the first little bit, you're not protected at all. And so therefore, we have to be careful because we heard people, I got the shot, and then what happened was that I got sick 6 days later. Well, that's too short. You haven't built up the antibodies. So therefore, getting the shot doesn't mean that you can get rid of the PPE and the physical distancing and all of those things. We still have to take care of that. Now, what about these percentages that we heard? Well, if you measured it right here in terms of these many people got sick and these many people got sick here, you would say this is maybe like 60% benefit, a reduction, right? But then if you come all the way out to here, you could see from this point, lots of people are sick here and only a few here. Then you could see that 94, 95% benefit would be there. So this group actually hasn't changed at all, right? In terms of the percentages, it's this group that's actually getting more infected. So that's why when you hear after the first shot, it might be 50, 60%. And then after the third shot or second shot, then it becomes this 95%. just remember this group looks pretty flat. So they haven't actually changed. It's the other arm that's actually getting infected that's driving the numbers wider and wider. So hopefully what this will mean is that you feel rest assured that if you've already had one shot, that you are protected. We still want you to do physical distancing and things like that to make sure that there's no new versions or anything that might be cropping up as well. Now, if you look at the AstraZeneca one, and I show you the graph very quickly here, you'll notice that this one is the chimpanzee version. So anytime you see CH and then AD, so that's a chimpanzee adenovirus. So that's using a virus to carry in the little piece of DNA to tell you how to make those spike proteins. So it's using it as a bus, as a delivery source. And what you see is the following, is that this one is not flat. So in other words, there are still people getting infected on this side. And so therefore you don't see a flat line going out here. So that might be why, there might be differences in some of the vaccines based on how they work, et cetera. So therefore, for the ones that you've all been given, which would be the Pfizer or Moderna version, we could see that sort of flat line happening. And so therefore you're protected. We don't have the adenovirus right now here in Canada yet. But hopefully soon in the future we might have. So you might see some differences there as well. And then we'll see how it works in different populations as well. But rest assured that duration difference right now for the 2 mRNA vaccines looks like it's going to be okay. So therefore, even if there's a small delay, there shouldn't be any harm that's coming to you and you're still protected. But you still have to do physical distancing, masks, you still have to do all of those things to make sure that you're completely protected. So I'll stop sharing my slides there, and hopefully that was useful, because I know this was reassuring to my friend when I started explaining all of this stuff to her in terms of these graphs, and hopefully it's reassuring to everybody else listening in.
Well, thank you, Peter. It really is kind of remarkable when you see those graphs and see the effectiveness of the RNA vaccines in particular. It certainly gives us a bit of light at the end of the tunnel here that we may be able to get past this over the next few months. So that was Dr. Peter Lynn, Director of Primary Care Initiatives at the Canadian Heart Research Centre. We're also really fortunate today to have a second expert, Dr. Carolyn Quash-Tan. And Carolyn is actually the chair of the National Advisory Committee on Immunization, NACI. This is the organization that all the provinces look to for guidance around the policies that they're setting currently for the administration of this vaccine and really all vaccines. So Carolyn, over to you.
Well, thank you. I think what Peter presented was absolutely great. They are the data that NACI looked at when we made our recommendation. I think that if we had enough vaccines, we wouldn't even have, batted an eye. We would have given it to everyone at the 21 to 28 day interval. But at one point in time, when there's scarcity of vaccines in terms of public health, you then have to ask yourself how best to roll out this vaccination program. So as Peter said, in both trials, participants got their second dose between 21 and 28 days, depending on the vaccine, up to 42 days. And that's how NACI said, well, at least we have some data to show that people could get vaccinated up to 42 days. And this overall, average estimate is a summary of all that. Of course, we don't have the data stratified by interval between the dose, but it's a start. We then looked at modeling, mathematical modeling, to see how best to have the most gains in terms of public health. And what those modeling show is that you are going to have much more gains in terms of decrease in hospitalization and death if you are able to vaccinate more people as the pandemic is roaring. So of course, if you live in Nova Scotia, when there's absolutely no case or barely any, you have the luxury to wait, to vaccinate everybody with 2 doses as the supplies come in. But if you're in Ontario and Quebec, where we've had more than 1,000 cases a day, the actual benefit will be much greater if we spread out that first dose to more people who are at risk, and then waited a little bit longer before giving that second dose. We were not expecting to have so much problems getting the Pfizer and the Moderna vaccines. I mean, as of January 25th, we've barely had any vaccines being brought into Canada. So that increases the difficulty. We were hoping to be able, to keep that 42 days moving. But just to say that all the two jurisdictions, so Quebec and the UK, who have had a 90-day interval, are currently doing surveillance of their vaccine effectiveness. And they've presented data to NACI this afternoon. It looks, that at 4 weeks, we still have good vaccine effectiveness. Of course, we will need to see how that plays out in the longer term. But I think, you have to realize that if you still have, let's say, 70% effectiveness at 4 weeks, and I'm just giving a invented number, it won't decrease to 0 the day after. So that decrease in vaccine effectiveness will be slowly decreasing. And hopefully, when you have a vaccine effectiveness and individual vaccine effectiveness that is lower, one would hope that at that point in time, the transmission in the community would be less, so that the gain would have already been gained. So I think that's the thinking that NACI went through when we made that recommendation.
Terrific. Thank you very much, Carolyn. So we've received a number of questions from healthcare workers in Ontario hospitals, and they're questions that I've also heard in our own organization. So I'd like to read a couple of these out and get a few thoughts for both of you on the best way to address those. So the first of these you've touched on a little bit, but I think this is the most common question that certainly we've heard. I've heard that vaccine efficacy has not been specifically tested beyond 28 days between doses. And the question is, who is monitoring this as we change it? And how can we be confident that it's really going to work as well without this clinical evidence?
The interval between doses for both trials was up to 42 days. So there is in there people who got it up to 42, even to 49 days with the Pfizer product. So as much as I can't tell you, at 42 days, your vaccine effectiveness is X, we know that overall, it's still 95%. Who is monitoring this is a great question. It's currently, because Canada is Canada, it's monitored at the provincial level. So that means that each province is following up their cohorts of vaccinees and looking at the date of vaccines and the risk of having a COVID PCR positive. So Quebec has already started to look at what has happened in the long-term care facilities, and those results will be published soon, I suppose. We know that BC is also doing that, and I would think that Ontario is doing the same thing. The Public Health Agency of Canada also mandated a new committee, which is a vaccine surveillance reference group that is co-chaired by Scott Halpern and myself, to make sure that all the gaps that could still be out there in terms of vaccine effectiveness and in terms of safety are actually pinpointed, so that there are projects that are put in place to be able to answer those questions. So vaccine effectiveness is something that is followed at the provincial level.
Great. And Peter, maybe if you could just touch on, where did the 21 days and 28 days come from? Why were the trials done that way to begin with?
Yeah, I mean, if you look at traditionally, this is when we would boost up with all of our immune system. So give your immune system time, and then show it again. And that way you get a better response. So that's why traditionally we tested around a month, like if you look at some of our hepatitis B vaccines and things like that, you could see that timing interval as well. And that's what they tested initially in their phase 1, phase 2 studies. And then they showed that there was very good antibody response. And then what happened was that they rolled out forward. And so we thought everybody would be getting it at 21 or 28 days as tradition. And then the supply issue came. Nobody expected the supply issue, because when the decision was made to spread it out, to actually vaccinate as many people as possible, we were reassured at that time that the vaccine would come. And so therefore the people where we use the second dose, there will be another dose coming to replace that second dose. And it was that reassurance. Then people said, okay, let's go for it. And let's spread it out to as many people as possible. And that would have been a perfect plan if the shipments had kept coming in. So when the shipments didn't come in, that's when people started to panic. So I think the key thing that people need to remember is that it may be that first dose is giving you enough antibodies. So let's say here's the magical line, and the first dose gives you up to here. Yes, you're right. The second dose will push you way up, but you don't need all that buffer cushion. So it's like having another bunch of zeros in your bank account if you're already rich enough. You've got enough to buy food, and I give you another $1 million. It'll make sure that it lasts longer, which is fantastic. But I don't think that you'll be left completely uncovered. So I think that was the worry that people said, well, if I don't get the boost, I'm just going to lose everything. It's almost like a rocket ship, like you need a booster to get you into orbit. And if I don't get that boost at the right time, I fall out of orbit and everything is going to go bad. But it looks like that first shot is doing quite a good job to give you enough antibodies so you stop invasive disease. So if the virus tries to come on board, it will not let it come on board and make you sick, which is really what people were looking for to protect people from getting sick. Now, having said that, I've been talking about, and that's the same thing with public health, and NACI has been saying, vaccine, no vaccine, it doesn't matter. You still have to do physical distancing. You still have to get mask. You still can't go and hug grandma, without a mask, right? So all the things that we need to do, we still have to do. So therefore, don't get frustrated. Everybody will still do the same thing to protect themselves from new strains, or maybe you're the 5% that didn't make the antibodies. So you'll still be doing the same things. Those physical barriers, the virus cannot get across. And so if we keep doing that, then the vaccine, even if it's sort of waning a little bit, you're still very protected. And then we'll give you a boost as soon as the new supplies come back in.
Peter, you had said it during your presentation, but as you said, in vaccinology, even if you don't find antibodies left when you measure, let's say for hepatitis B, you still have memory cells. So that boosting effect will still come. So it's not because you can't find anything that you're still not protected. Those memory cells are still there. And as Peter said, there's something to give it too early, but there's absolutely no data that show that a longer interval is going to be detrimental. And I think people need to also remember that.
I guess that's a very good point, right? Because the original study was to say, how fast can I give it to still get an effect of that boost? And that's why they tried to keep it, 21 days, 28 days. In other words, can I just do it and then do it quickly again to get the maximum boost? But in fact, if you look at all the other vaccines, if you pull it out, that second dose pulls out, you actually get a better response, right? So it was more like, how quick could we do the 2 shots in people? As opposed to, oh, if I miss that 21 days, I'm not going to get a boost at all. So I think people need to understand that originally how fast could we do it, in terms of squishing it together, as opposed to if I miss that window, then I get no effect at all. In fact, if that window is out a little further, you actually get a better boost with all of the other vaccines that we've seen before.
Yeah. In fact, there was some data released earlier or very recently that the 12-week delay that is being practiced in the UK for their adenoviral vaccine, they saw even better responses at the 12-week time frame. So, but that doesn't change the fact that our clinical evidence is based on the clinical trials, and that's certainly what we'd like to be able to do. So the next question I've also heard, and it says, can I mix the vaccines by getting 1 dose of each? So can I get Pfizer and then come back 3 weeks later and get a Moderna? Right, Peter, we'll start with you this time.
Well, Carolyn can go ahead because there's an official position, and also there's studies now ongoing to try and figure this out. Carolyn, go ahead.
Sure, thanks, Peter. So the official guidance is that there's no data on interchangeability. However, because those are mRNA vaccines with, protein S, spike protein that are quite similar in their conformation, we've said that if you don't know what you got, or if you run out of doses, you could use the same platform to give that second dose, but again, with no evidence, but it just makes sense. As the mix and match of AstraZeneca plus an mRNA vaccine, the UK has started a study on February 1st, and we'll see how that goes. But a study that is in preprint status on medRxiv shows that in mice, at least, this mix and match actually seems to give a better T-cell response. So I think there's something there to be said, but we need more data before making an official recommendation.
Peter, so for you, if my second dose is delayed longer than 42 days, do I need to start over again and get my first dose?
Yeah, so the good news is no. So in the past, when we have a delay in dosing, let's say for other vaccines, we notice that the antibody boost is good. So in this particular case, there's no need. And then also what we're going to do is you're going to still practice all that physical distance and everything else. And some of the companies are saying with new strains coming on board, there might be a third boost that they were talking about, so that we give you even more money in the bank, so to speak. And so therefore, you don't have to restart the whole series all over again. If you're at 42 or 50 days or 60 days or even 90 days, most likely you will still be protected as well. So I think that one is something that people were worried about, that if I miss that window, I'm going to have to start it all over again, or I get zero results because there's no benefit at all, which that part is probably not true. As you can see by the graph, it's not like after the second dose is the only time that you're protected. You're already protected from the first dose. The second dose is just giving you more antibodies to play with.
And nothing different between the doses. The first dose is the same thing in the vial as in the second dose, right?
Exactly. Well, you have to squeeze it out of the vial, right, to make sure you get that sixth dose coming out of there.
Carolyn, how can I trust the vaccine that's been developed so quickly? So we've heard a lot about how fast this got approved and how fast it got tested. I think there are people who sort of worried that maybe corners were cut, or this was accelerated in some way. What's your response to that?
Well, I think the studies were well done. The difference is that, usually you do your phase 1 and then you look at your results, you try to come up with enough money to invest in your phase 2. That usually takes like 12 to 18 months. So the phase 1, 2, and 3 were done exactly the same as usual. It's just that the in between phases were shortened because governments put money into these companies and there were investments from various people. And everybody was ready to launch, and so recruitment went quite well. The only difference is that the phase 3 trial, which usually should have lasted 12 to 18 months and sometimes up to 2 years, was not stopped, but the data were looked at an interim point when they had enough outcomes. So what that means is, in terms of efficacy and safety, they had very good data for the first 2 doses up until 14 weeks for the two mRNA vaccines after the second dose, enough to have your 7 days where you look intensely at adverse events in people after each dose, and the month after where everything that goes wrong needs to be reported. That's always the same. The only thing that's missing is whatever happens to people over the next 1.5 to 2 years. If they are admitted or if they have any problems, then that should also get reported. We usually don't tend to find that many things in the last year, 1.5 years, but that is still ongoing. Health Canada did not give a full authorization. They gave an interim order asking the companies to continue with the follow-up. So that's why we've had data so quickly. The other difference is that usually Health Canada has set times at where companies can submit their data. Whereas because it was a rushed, what they said is, every time you have data, just send them in on a rolling basis. And so they've been working on these data continuously. I think people have to realize that we've all worked day and night on those data. Health Canada has been pulling in resources and has been very diligent at looking at data, and NACI has been working more than ever. Usually it takes us like 1.5 years to spit out a recommendation. Now we've done, 3 to 4 in the past 2 months, and every time there's a new product, we're at it again. So it's more people working on this and more investment that allowed the studies to be done not more quickly, but with less lag between phases.
Yes, and in fact, I think if anything, this vaccine or these vaccines and this disease probably has more attention than just about any vaccine or any disease has in history. This includes the regulatory bodies here in Canada, but also independent regulatory bodies around the world. And if there's any advantage of things going a little slow in Canada, we can take solace, I think, to some degree in the fact that tens of millions of people have received this vaccine around the world, and there's a lot of monitoring and a lot of studying going on, and so far everything reads out as these being extremely safe vaccines.
The media has been scrutinizing every single case. So if there's a Bell's palsy, it's reported on the media, for example. So in a strange way, the microscope on the vaccine is so powerful. You've got the media reporting, you've got all the health folks looking at it, the regulatory agencies, you've got people like NACI, you know, thank you, Caroline, for looking over the data and making recommendations. So everybody's looking at it. So if anything, this is the most well-studied area that we've ever had in medicine, right? So yes, it's in a short period of time, but you had a lot of eyeballs looking at it, and nobody wants to send out anything that can cause harm. And that's really the most important thing, is that nobody involved in this wants anybody to be harmed. So that's why everybody is keeping a very close watch on it. So rest assured that the eyeballs, many eyeballs, are on the data and on all of the studies and on all of the real world data that we're collecting right now. I mean, so we're seeing from Israel, we're seeing lots of numbers coming out, and they're telling us that it's working, you know, like people aren't getting sick when they have the vaccine. So it's very reassuring, actually.
Okay, final question to you, Peter. And you touched on it a little bit on your introduction, and the question is, am I still going to have to wear PPE after I've been vaccinated? And, you know, even looking at your hockey stick, your inverted hockey stick, if I'm 2 weeks out, I'm on that flat part, am I okay? Or what's your advice?
Good question. So the bottom line is that you're still going to do PPE, you're going to still do physical distancing for a couple of reasons. So the number one reason is to protect yourself. So what happens if you're in the 5% or something that doesn't make antibodies, right? We know that there are people that don't respond to vaccines. So in hepatitis B, not everybody responds to the vaccine, doesn't make antibodies. So therefore, could you be one of those? So that's why we want to still keep the physical distance, et cetera, to make sure that you don't happen to be one of those people. Number two is, if there's a new strain that comes out, we're not 100% sure that the vaccines will work. So if the spike protein changes or something like that, then we're not 100% sure. So therefore, as frontline workers, you might be exposed to people. So again, physical distance, masks, shields, all of those things still need to be in place. And then finally, remember, these things are meant to create antibodies, which are very good at patrolling your body. So stopping that invasive disease, right? But it's not very good at getting rid of the virus if it's in your nose cells, quietly photocopying itself, not invading anybody. So in other words, people that are asymptomatic carrying the virus, we're not quite sure if the vaccination would get rid of the viruses in the nose, for example. So we don't want you to accidentally say, I'm vaccinated, so there's no way that the virus is in my body, and then go and hug grandma and accidentally bring the virus to grandma. So even if you're vaccinated, we still want you to do the physical distancing, do the PPE and the mask and shields when you're at work, and also respect those same borders with your family and loved ones, just to make sure, until we sort this out, can we make sure we get rid of all virus in your nose? Or is it just protects you from getting the invasion, in other words, the illness? Because if it's only the illness that it gets rid of, you could still be a carrier of this virus, even though you're vaccinated. So that's why we still take the same precautions. So for the people that haven't gotten a shot, don't feel bad, because the people that get the shot, they still have to do the physical distancing and they can't go and hug grandma yet. So therefore, there's no harm done. So everybody, let's just play by the same playbook. Because remember, no matter how much this virus changes, it cannot grow wings and it can't grow feet. So it can't get across physical barriers that we put in place, the distance, the mask, the ventilation, you know, and short duration of contact. Those things the virus can never get around. So let's just make sure we use them, whether you get vaccinated or not.
Oh, terrific! You know, and it also, as we've all seen this year, protects you from other infectious diseases. It's the first year in my life I've never had a cold, and in the hospital we haven't seen any influenza at all this year. So this clearly is effective at preventing transmission. We're gonna end the questions now. I'm gonna give Peter and Caroline, each of you, just another minute or two if there's anything else you'd like to leave.
In the end, just to rest assured that NACI is monitoring all the new evidence, and as data come out, we are going to make changes to our recommendations. Surveillance is ongoing. The provinces are very well equipped and prepared to do both safety and effectiveness surveillance, and those data will be available in a short time. So if we see that there's anything that changes in terms of, let's say, 90-day interval would be better, then we would make that recommendation. But at this point in time, we're just looking at the data, and we really try to make evidence-based decision, keeping in mind that we also need to take into account ethics, equity, feasibility, and acceptability.
Yeah, and I want to thank Carolyn for looking after all of that so that she can make the NACI can make these recommendations to us. And I want to thank all of you that are listening for doing your jobs and doing it brilliantly over this time period. And I know that you're putting yourselves at risk. And rest assured that when people say 42 days or 90 days, there's some science behind it. So they're not sacrificing your safety, because I know that was the first belief that people are saying you're just making up numbers. There's nothing to back that up. So remember that everybody wants you to be safe, because if you're safe, then you can look after patients. And so that's why NACI, the health, the Health Canada, everybody that's reviewing this data is trying to make sure that people are safe with these vaccines. And number two, that they will get the protection. Having said that, the vaccine, as we heard, the viruses are going to be changing and things like that. So remember, if you get a vaccine, it doesn't matter. You're going to do the PPE, you're going to do the physical distancing, make sure that the ventilation is good, and make sure your contact is short. And let's just keep doing that, because we know that that will keep the virus at bay. And for those that are awaiting your vaccines, continue to do that. So basically, our game plan is exactly the same. It sounds very simple, but it is a very effective way of keeping this virus away. And as people get vaccinated, rest assured that the science is behind you. And our goal is to really protect you, because by protecting you, you end up looking after all of us. So we want to thank you for all of that. And I want to thank you, Brad, for moderating this. That was beautifully done. And Caroline, again, thank you for all the work that you're doing at NACI. I know that you've spent many nights, reading things and analyzing things and doing that on our behalf. So we really appreciate all your efforts.
Peter.
Well, thank you once again. Thank you, Peter. Thank you, Carolyn. And thanks to all of you that are tuned in today. We hope it's been helpful. And we are certainly grateful to all of the hospital staff for their continued hard work and commitment to delivering the best possible care in what are really truly extraordinary circumstances. So to everyone, continue to practice your social distancing, wear your PPE, get in line for the vaccine when it's your turn, and stay safe. Thank you.